Hi everyone, in this video we're going to be looking at the rest of the digestive system, focusing on the small intestine and ending with the large intestine. The learning objectives associated with this topic will be specifically looking at this red box at the bottom. As ever, please make sure that you read the textbook pages outlined above and you complete the booklet pages of your notes. These are the key words associated with today's lesson. There are a lot of new words that you may not have heard before, so it's really important that you do read your textbook and you do understand what these words mean. The small intestine is called so not because it's short, but because of the diameter. The small intestine is actually about 6 meters long. The first part of the small intestine, which you can see here that connects from the stomach, is called the duodenum. It's about 25 centimeters long and it's recognizable in being the first part of the small intestine. As food enters the duodenum and it enters the rest of the small intestine, where we have the jejunum and the ileum. This is where the majority of complete digestion and absorption of nutrients takes place. It does so with the aid of the accessory organs, the liver, gallbladder and pancreas, and also with its own secretions of juices that aid digestion. Within the small intestine itself, we also get mechanical digestion, and this occurs through a process of segmentation. So segmentation relies on peristalsis. So remembering that peristalsis is the contraction and relaxation of smooth muscle along the length of the intestine. And what that does, as you can see in these diagrams, is it gets that food bolus and physically breaks it up into smaller and smaller pieces. So we still have the same chemical composition, but we have smaller smaller particles, which leads to a larger surface area to volume ratio. What's important to understand about the small intestine is that the pH is changing. If we look at this cartoon picture here, we can see that in the mouth, the pH is relatively neutral. As we swallow food down the esophagus and it lands in the stomach, the pH dramatically drops and it becomes about 1.5 to 2.0 because of the hydrochloric acid that is secreted from the gastric pits in the stomach. If you've forgotten what's happened in the stomach, I would encourage you to revisit the previous lesson and to look at your notes. After food has been in the stomach for between 2 to 8 hours, it then leaves through the bottom part of the stomach into the duodenum. And we can see here that the pH starts to rise quite rapidly. We've gone from 1.5 to 2.0 to somewhere between 5.6 to 8.0. What I need you to note here is that the pH of the duodenum starts to become alkaline. So in the oral cavity, that is the mouth, we are neutral. The stomach is very acidic and the duodenum is alkaline. We can see here on the pH scale that 0 to 6 is considered to be acidic, with 1 and 0 being the most acidic. Number 7 is neutral, and anything above 7 is alkaline, with 14 being the most alkaline and 8 being the least. Associated with the small intestine are the accessory organs, as I've mentioned before. Pancreatic juice and the bile that is stored in the gallbladder plays a really big role in digestion here. The pancreas is regarded as an accessory organ because it's not directly part of the alimentary canal, although you cannot survive without it. What the pancreas does is secretes digestive fluids directly into the duodenum, which you can see through this little duct here. The fluid that enters the duodenum, that's the first part of the small intestine, is alkaline, and crucially what this does is it neutralizes the stomach contents. So remembering that the stomach lines itself with mucus to protect from the acid, well, the rest of the alimentary canal doesn't have that protective mechanism, so it has to rely on something different, which is this alkaline fluid to neutralize that acidic chyme. The pancreas also secretes many enzymes that help to completely digest the food nutrients. The ones that you must know about are pancreatic amylase. We call that pancreatic amylase to differentiate it from salivary amylase in the mouth. And what this does is it breaks down starch into smaller disaccharide molecules. The next enzyme that we have is pancreatic protease, or sometimes known as trypsin. This will digest proteins into peptides, which is very similar to what happens in the stomach. And the last one is pancreatic lipase, which breaks down fat molecules into fatty acids and glycerol. So now we know that the pancreas produces digestive enzymes and secretes secretions to help neutralize the acid that comes from the stomach. Now we turn our attention to the liver and bile. What's important to know is that bile is produced in the liver up here, and you can see that these tubes transport that bile duct into the gallbladder. The gallbladder stores bile, so bile is produced in the liver but stored in the gallbladder. Bile plays a really important function in the body. It helps to emulsify fats, which we're going to talk about in a second. So bile is not an enzyme, it's an emulsifying agent. Emulsifiers such as bile actually break fat down into smaller droplets. So they don't change fat into a different chemical, so therefore this is regarded as mechanical digestion. And again, the reason why this is so important is because it provides a larger surface area to volume ratio for fat to be digested. Bile is secreted from that gallbladder and it goes down this bile duct directly into the duodenum. This cartoon diagram simply shows the emulsification of lipids. So what we can see here is a large fat globule, and as it enters the small intestine, bile is secreted from the gallbladder through that bile duct, and what it does is it breaks that fat droplet up and it sticks to the fat droplet. So this bile salt here will break up the fat, and it will stick to the outside of that fat droplet. And because it has a hydrophilic and a hydrophobic side, it can stick to that fat droplet really easily. But what's really important about this is that the hydrophilic sides are facing each other. And because like particles repel, it prevents the fat from joining back up and forming a large globule again. And this is how that large surface area to volume ratio is achieved. So just to give you an understanding of what happens in each part of the body. This is looking at proteins, but this could be true for any enzyme. So here we have a large macromolecule, which is protein. This is chewed, which is aided through mechanical digestion, and it's swallowed into the stomach. The hydrochloric acid will denature that protein, and it will change the physical structure, but we still have one large protein molecule. Pepsin, the enzyme found in the stomach, will actually start to chemically digest this large protein molecule, and it breaks it up into smaller chains of polypeptides. Once this protein polypeptide leaves the small intestine and enters the duodenum, first part of the small intestine, it will start to be broken down further until eventually we reach the final stage of digestion in the small intestine, and the polypeptide molecules have been completely broken down into single monomer units, which are known as amino acids. So a summary of what we've talked about so far is that the duodenum is the first part of the small intestine. It's about 25 centimeters long, and what happens here is alkaline digestive secretions from the pancreas enter from the pancreatic duct into the duodenum, and this changes the pH environment from acidic to neutral or slightly alkaline. The pancreas also secretes digestive enzymes to further break down carbohydrates, lipids and proteins by chemical digestion. The liver and bile, which makes bile, is stored in the gallbladder. But the bile is secreted into the duodenum to emulsify fats. Emulsification is a form of digestion, and it's mechanical digestion, where large fat droplets are broken up into smaller globules to increase that surface area to volume ratio. This will mean that lipase can break down lipids into fatty acids and glycerol at a much faster rate. In addition to secretions from the pancreas, the intestinal glands, which is the lining of the small intestine, will secrete intestinal juices. Now, these intestinal juices will complete the chemical digestion of all the foods that we have eaten. So that is to say, they will completely digest carbohydrates, lipids and proteins. The intestinal juices will secrete a collection of enzymes called amylases, and these intestinal amylases will break down the disaccharide molecules such as maltose and sucrose into very simple monomer sugars such as glucose and fructose. Peptidases, which are protease enzymes, will further completely digest peptides or polypeptides into amino acids, while lipases will further digest lipids into fatty acids and glycerol. Here is a summary table of what's happened at each stage in the alimentary canal. First of all, if we look at the mouth, we have the substrate starch, which is digested by salivary amylase into maltose. In the stomach, we have the substrate protein, which is broken down by pepsin into polypeptides. While in the small intestine, we have the pancreas, which secretes its pancreatic enzymes, which are pancreatic amylase, pancreatic protease or trypsin, pancreatic lipase, and ribonuclease or deoxyribonuclease enzymes. You can see here that their substrates very much mirror the names that they come from, and these are the end products. It's important that you take the time to remember and understand the names of these enzymes, the substrate, and the end products produced. Finally, in the small intestine itself, so here we've just shown the duodenum, polypeptides are completely digested to amino acid by peptidases. Sucrose and maltose, which are disaccharide molecules, are completely digested into monomers, such as glucose and fructose. And we still keep working on the digestion of fat into fatty acids and glycerol. Okay, so this diagram is just a similar summary of what we've been talking about so far. Please make sure that you stop the video here and you read this information carefully so that you understand how digestion fully occurs. So now that we understand how food is completely digested into its monomer units, we need to now look at the small intestine structure and function. If we look at a cross-section of the small intestine tube here, we can see that the inside lining is made of these finger-like projections, and they line the entire small intestine 6-meter tube. So in this picture here, we can see each individual one, and here we have a schematic diagram to show how that looks. So the adaptations of the small intestine are all geared around absorption of nutrients. We need to absorb these nutrients so that we can get them into our bloodstream and then into our body cells. So the adaptations that we're going to be looking at are, first of all, the small intestine is very long. The lining, which is the mucous membrane, or is folded. So we say it has a folded mucosa. Each of these folds are known as a villi, and each villi has microvilli. The small intestine has a very unique structure that specifically suits its function in absorption of nutrients. The first thing that we need to look at is inside the small intestine. It has these finger-like projections called villi, as we talked about, and what makes them very unique in this instance in being able to absorb nutrients rapidly is its large blood supply. So here we have an image of a villus, and we can see that it has a very large blood supply inside, and this will allow for nutrients to be rapidly absorbed through that mucosal lining, through those epithelial cells. So once we've had digestion in the small intestine, these monomer units, such as glucose, amino acids, water, and water-soluble vitamins, will be absorbed directly into the capillaries. The nutrients will then go to body cells and to the liver to be detoxified. If we look at the center of the villus, if we go right here, we can see that this structure is a lacteal. Now you understand from previous lessons that a lacteal is part of the lymphatic system. It's a blind-ended duct that absorbs the nutrients that are being produced by the products of fat digestion. So fatty acids and glycerol will be absorbed through that epithelial lining directly into the into the lacteal, where these fatty acids and glycerol will reform, and they will move throughout the body via the lymphatic system. Eventually, those fat products will return to the blood capillary and the blood vessel system, but that won't happen for a little while. This diagram can be found in your textbook, and it's really important that you stop here and that you read this information carefully. Things that you need to understand is that the villus has a very specific structure which is related to its function. Here we're telling you that each villus is about 1 millimeter long, and we have a capillary and a lacteal, which is surrounded by that capillary network. So absorption of nutrients through that 1 cell thick wall allows this absorption of nutrients to happen rapidly. And here we are talking specifically about the mode of transport. Simple sugars such as glucose are absorbed by active transport, and they pass through the cells on the outside of the villi into the blood capillary. So this is different from what happens inside normal cell structures, which is facilitated diffusion. The type of transport to get glucose from outside of the the villus into the blood capillary is active transport. So now that we've looked at the small intestine, we're going to just look at the last stage of the alimentary canal, and this is looking at the large intestine. So if we just focus on the anatomy for a second, we know that we have an appendix. Some people may not have that anymore. It could be removed if it becomes infected. But we have the ascending colon, the transverse colon, and the descending colon. Now these are all parts of the large intestine, which ends in the rectum, which is where feces are stored, and then we have the anus at the bottom, where feces are eliminated from. Now, the large intestine is called so because of its wide diameter, but it's actually shorter than the small intestine. It's only 1.5 meters in length. The different parts, as we've discussed, are the cecum, the colon, the rectum, and the appendix. And you can see that in this diagram over here. There is no chemical digestion that happens in the large intestine. We only have mucus secretion into the lining of the large intestine. Typically what happens here is water absorption. So this is really important that you understand that water absorption is the main process of the large intestine. Bacteria is found here that helps to further break down these organic compounds that have made their way through here, and it converts them into the vitamins which are reabsorbed into the body. Anything that is undigested will become feces. These are food materials that have not been digested. Bacteria and bile pigments, dead cells and water. As this food, which is undigested and then becomes waste, leaves the body, we call this defecation. Okay, so this type of removal from the body is not excretion, it is elimination. And it's really important that you understand the difference between elimination and excretion. So elimination is the removal of unwanted, undigested materials. Excretion is the removal of metabolic waste. So the diet can affect how the large intestine works. It can lead to conditions such as diarrhea, bowel cancer and celiac disease. So it's really important that we understand about our digestive system and look after it. This is a video which will help you to understand what goes on in the large intestine. I recommend that you watch this on the PowerPoint. And here is a summary of what we have learned so far. So we know that the small intestine secretes intestinal juices that contain enzymes to completely break down food into its monomers. We also know that the small intestine has many folded structures called villi, and these greatly increase the surface area to volume ratio for rapid absorption of nutrients. Each villus is only one cell thick. We saw that in those diagrams before. So the epithelial lining is only one cell thick, which allows for rapid absorption of nutrients. Each villus has a large blood supply and a central lacteal. There are also microvilli found on the surface of the villi themselves, which further increases the surface area. Fatty acids and glycerol are absorbed directly into that central lacteal, where they will become lipids again and be transported around the body through the lymphatic system. Glucose, water, vitamins, minerals and amino acids are all absorbed into the capillary within the lacteal to then be transported to body cells. The large intestine is only 1.5 meters long. It's short in length, but it has a large diameter. In the large intestine, no digestion occurs. However, the function of the large intestine is to absorb water back into the body and to break down organic compounds and convert them into vitamins, specifically B and K. The resulting feces that we have in the large intestine are stored in the rectum and eliminated through the anus. Diet can have a significant effect on the health of the digestive system and can lead to conditions such as diarrhea, constipation, bowel disease and celiac disease. Here we have a cartoon summary of what is going on throughout the entire digestive system. I would strongly recommend that you take the time to look at this and perhaps create your own diagram to help you revise and understand what is happening at each stage. Color coding would be useful here. If you need a different perspective, this is a really useful video from Crash Course that I would recommend that you watch. You can watch that in your own time. And finally, tasks that you need to complete are pages 15 to 19 of your booklet. And as ever, I strongly encourage you to read your textbook, as this contains far more detail than what is found in these PowerPoints, and it will help you to understand the detail that you need to know for the exam.